Earth is vibrant with life. From deep oceanic crevices to extreme climates, life is at least present in the form of microorganisms. Cellular organization happens at a higher level, forming complex plants and animals. The internal processes of living beings are controlled by stringent metabolic pathways forming delicate networks composed of several genes and their regulators. The origin of life is basically the story of how several molecules came together and organized themselves. The story is quite complex, and we still have not moved past the prologue. This video is just an in-depth introduction for researchers, students, or maybe just a science enthusiast. Evolutionary biologists Carl Woese, Otto Candler, and Mark Willis created a three-domain system and divided cellular life into archaea, eukarya, and bacteria. We can create a phylogenetic tree to describe how simple life evolved into separate organisms. And if we tune back, we can imagine an organism, a very simple one, from which everything originated. This is called LUCA, or the last universal common ancestor. You can think of this as a germinating plant having three leaves corresponding to the three domains, and the seed from which everything originated as LUCA. Unfortunately, we cannot find any living LUCA now, nor are there any fossil records of it. This is just a hypothetical simple organism containing nucleic acids and proteins. When it comes to a discussion about the origin of life, we can try breaking it down into three important questions: how, why, and where. Also, the answers to when and what are still kind of ambiguous, but scientists have found signatures of life approximately 4.1 billion years ago in the form of carbon-12 isotope in zircon, which is basically a mineral. On the other hand, scientists and philosophers have their own opinion on what is life. But what we can do is we can detect if something is living or not based on certain characteristics. Like, is it responsive to the changes in its environment? In other words, is it aware and can it maintain homeostasis? That is, can it regulate its internal environment taking cues from the external milieu? Can it metabolize? Or simply, can it produce energy and other chemical intermediates from its food? Can it reproduce? That is, make duplicates of its genetic material? Now obviously there are other features describing life, but I think the clue lies in clearly understanding these four words. Aristotle basically had an idea that in scientific terms is now known as the spontaneous generation. That is, life originated from organic matter, just spontaneously by chance. For example, flies originating from rotten waste. This theory, however naive, was supported by scholars even until the late 17th century. In 1668, Italian naturalist and the father of experimental biology Francesco Redi proved that maggots do not originate in meat unless flies are allowed to lay eggs on it. But Louis Pasteur in 1861 put the final nail to the coffin by proving that microorganisms do not arise in sterile nutrient-rich media kept secluded from the environment. After Pasteur's experiment, spontaneous generation was completely disproven. Also came along Darwin with his Origin of Species and his theory that complexity evolves from simple life. This is a very interesting and probably the first true scientific theory for the origin of life. Russian biochemist Alexander Oparin and British Indian biologist J.B.S. Haldane postulated that life originated through molecular or chemical evolution. They hypothesized that primordial Earth had a reducing atmosphere composed of methane, ammonia, carbon monoxide, water, and so on, with little to no free oxygen and ozone. This oxygenless environment catalyzed the formation of simple organic molecules, which were diluted in the primitive oceans, forming a primordial soup. These simple organic monomers and lipids again had combined due to electrostatic forces to lead to a liquid-liquid phase separation, causing coacervate droplets. These droplets would then fuse with other droplets or form more droplets upon fission. Therefore, Oparin and Haldane conceived an idea of an extremely primitive type of cellular metabolism. The primordial soup hypothesis established the foundation of abiogenesis. In 1952, Harold Urey and Stanley Miller at the University of Chicago conducted an experiment which went on to somewhat prove Oparin and Haldane's primordial soup hypothesis. It was a complex experiment, but the basic skeleton of the experiment was designed as such. A gas chamber containing methane, ammonia, and hydrogen mixed in a ratio of 2:2:1 was connected to a flask containing water. The water vapor that was generated on boiling would then pass to the gaseous mixture and was simultaneously subjected to continuous electric sparks. There was a condenser placed below the gas chamber which would collect the liquid obtained upon cooling down of the mixture. They performed paper chromatography, which was then a very powerful technique to identify and separate dissolved chemical substances, to identify the components of the mixture, and established the presence of glycine and alanine. Later, the samples obtained from the 1952 experiment were reanalyzed using more precise techniques, and other amino acids like aspartic acid, valine, etc. were also found. This was such a groundbreaking experiment that it was modified and performed later by several biochemists. Recently, these coacervates have gained some traction as protocell in the scientific community. Panspermia comes from two Greek words, pan meaning all and sperma meaning seed, and the name is quite self-explanatory. The seed of life is there, present among asteroids, space dust, and so on, and life originated on Earth after some meteorite carrying the seed of life bombarded Earth billions of years ago. Now there are some variations of the old theory. A new theory known as pseudo-panspermia suggests that organic molecules which originated in some place of the universe were brought to Earth by comets, and then these organic molecules formed life through abiogenesis. There is also another variation stating that the organic molecules originated in space and reacted chemically with one another, forming life somewhere in the universe, and that life was brought to Earth by comets. The former is more acceptable in the scientific theory. In 1969, a meteorite showered on Murchison in Victoria. Named after the city, the Murchison meteorite was analyzed by chemists, and organic molecules like some amino acids and even nucleobases, which are found in your DNA and RNA, were found. The meteorite Tissint, which came from Mars, also contained organic molecules. Therefore, the basic concept of pseudo-panspermia, that organic molecules can fall on Earth from other parts of the universe, was kind of proven right. The story is nowhere near completion. It does not make sense for organic molecules to react with one another. I mean, an amino acid or a lipid chain is useless without context. So now we need to bring in the two major players, the story of energy and the story of environment, into our story. We need energy to survive, and so did the most primitive life form. The energy currency of the cell is adenosine triphosphate, commonly called ATP. The modern cell has a stringent mechanism to monitor the rate of metabolism of ATP, and it needs to utilize the ATP for converting food into usable chemical energy. Erwin Schrödinger, in his book What is Life, wanted to connect the dots between the origin of life and free energy. It is necessary for life to increase the orderliness of highly disordered molecules. For a cell, even the simplest one that you can imagine, some energy is spent continuously to maintain homeostasis. That is, it needs to maintain an optimum concentration of molecules inside their membrane. Now let's take a small digression and talk about the two transport mechanisms of a cell by which it can let molecules enter or exit. One is passive diffusion, where no energy is required and the molecules move along the concentration gradient via diffusion. The other one is active transport. Energy is required and the molecules move against their concentration gradient. And the cell has specialized proteins that span the phospholipid bilayer and facilitate this transport. Therefore, the cell uses the second law of thermodynamics magnificently to maintain itself. Again, coming back to the story of the origin of life, undoubtedly maintaining this perfect balance is extremely important for the cell, and all the theories stumble answering the development of a transport process in the primitive life form. Suppose we have a lipid coacervate having a nucleic acid and some primitive form of polymerase and other enzymes, a perfect condition for replication. But it cannot replicate unless it has ATP, and having a proton gradient is absolutely necessary for generating ATP. Peter Mitchell proposed the theory of chemiosmosis to answer the formation of ATPs inside the cell, specifically the mitochondria. In simple terms, the cells undergo a metabolic cycle called the citric acid cycle. Ultimately, it generates an electrochemical gradient due to different concentrations of proton across the cell membrane, higher outside and lower inside. The differential concentration is formed due to certain enzymes in the membrane pushing protons outside the cell via active transport. Subsequently, the enzyme ATP synthase then allows this proton to diffuse into the cellular matrix and in turn uses the free energy to convert adenosine diphosphate to adenosine triphosphate. Now, as you can probably guess, this process of printing energy currency is super complex. But a cell has to undergo this because it needs the money. Even the most primitive organisms utilize proton gradient to produce ATP and in turn reduce carbon dioxide into methane, which is a hydrocarbon. Nevertheless, the necessity of a chemical gradient becomes super conspicuous. Let's stay here for some time and try to understand how primitive life might have solved the problem of chemical gradients. Probably the most important candidate for answering this is the deep sea hydrothermal vents. These are cracks on the seabed and are simply the hot springs of the ocean floor. They supply minerals from the Earth's interior into the ocean, thereby maintaining chemical balance. There are two types of hydrothermal vents, the black smokers and the white smokers. The black smokers, which are super hot and have a temperature of more than 300 degrees Celsius, have been disregarded as a potential candidate. And the white smokers, lovingly called the lost city, with a temperature of around 70 degrees Celsius, have become the primary stakeholder. Gunther Waxterhauser hypothesized that the hydrothermal vents might hold the secrets to the origin of life in his iron-sulfur world theory. Hydrothermal vents are an example of non-equilibrium dissipative system. I won't bring in too much physics and statistics, but in layman's term, the vents are the external source of energy and can help cross the free energy barrier during forming organic molecules. I have a plan on doing a series on implementation of basic physics and mathematics in answering biological questions. I might do some videos dedicated to thermodynamics and the origin of life. If you want the same, please let me know down below in the comments. The ocean has a relatively lower pH than the vent fluid. That is, the concentration of proton is higher in the ocean. Therefore, it is hypothesized that the vents might have acted as an inorganic membrane with a potential to transport protons from the low pH ocean water into the high pH vent fluid. And some primitive molecular machine has simply started to utilize this free energy. Researchers have also attributed the process of serpentinization, which is basically the conversion of iron magnesium silicates into serpentinite, for the origin of hydrogen ion gradient. It is a geochemical process and the hydrothermal vents are one of the places where this takes place. This can create hydrogen, for example during the oxidation of ferrous to ferric iron with sufficient reducing potential to form methane and small hydrocarbons. There are many complex pathways undergoing in the vicinity of the hydrothermal vents that can reduce carbon dioxide into methane and acetate. And guess what? This is gold for the primitive methanogens and acetogens. Dr. Nick Lane, a biochemist and evolutionary biologist from UCL, is a pioneer in researching how the primitive life form tried to tackle the energy equation. He juxtaposed the huge potential of hydrothermal vents and the primitive carbon fixating acetyl-CoA pathway, which is a central hub for metabolism inside the cells. This is also known as the metabolism first hypothesis. So we get a brief idea on how energy is necessary for cells and how the conditions of primitive Earth led to solving the energy mystery during the origin of life. Some of the most important theories on the origin of life. The theory of spontaneous generation fell spontaneously when Urey, Pasteur, and others showed that life cannot originate out of nowhere. Therefore, Oparin's model, Panspermia, and theories related to hydrothermal vents became more acceptable. They are not flawless, obviously, but still strengthened our understanding of the origin of life. The basic problem with all these theories boil down to homeostasis. Maintaining homeostasis is not a joke. A cell has thousands of specialized proteins called transporters that specifically let some molecules inside from a pool. These are just like doors, and not only that, they are highly guarded. Therefore, sneaking in is prohibited. As we also saw, this transport is necessary for a cell to produce energy. And without energy, a cell is dead. Moreover, not only transport but selective transport is necessary, even for the simplest of cells. The Miller-Urey experiment, which proved that essential organic molecules can generate from simple gaseous mixture, is not very true. There are some major flaws of the experiment. 1. The mixture of methane, ammonia and hydrogen that they used is now considered to be more reducing than what geologists claim the prebiotic atmosphere to be. Later experiments with different gaseous composition did not give very promising results, and models show that it is quite impossible to get a sufficiently concentrated mixture of life-building molecules. Also glassware. Urey and Miller used borosilicate glass, which might have acted as a catalyst for the reaction. But this also has a bright side. It can indicate that silicate rocks played a vital role in the formation of prebiotic life. One major flaw was the amino acid soap formed was racemic. It is a bit more scientifically challenging concept. So some molecules have chirality, that is, they can either rotate polarized light clockwise or anticlockwise, and they are termed as the L and D form respectively. In nature, we mostly encounter L amino acids. Therefore, there is some chiral selectivity among the basic building blocks of life, and none of the theories can actually explain that. Panspermia to me is an excuse. It may be enough to fertilize the earth and bring the first baby life, but we still have not answered the question, how did life originate? It is escapism. Also, according to this theory, the universe should be ideally booming with life, but as of now we have not found any signature of complex life anywhere else. Some recent theories are the RNA world hypothesis, proposed by Alexander Rich and Walter Gilbert in the 1960s. Basically, due to wet-dry cycles in prebiotic Earth, some nucleoside precursors and ribose sugar held hands together and formed an RNA. But inside cells, formation of nucleotides is not as easy as it sounds. Anyway, RNA can self-replicate. So now you have your primitive genetic material which can also act as an enzyme called the ribozymes. Now we all have catalytically active ribozymes inside our cells. To function, RNA needs to undergo conformational change called folding. Now, although ribozymes are perfectly capable of replicating unfolded RNA, it failed to replicate folded RNA. Therefore, ribozymes which have a specific secondary structure cannot replicate itself. Interesting paradox, isn't it? So one reason can be that the primordial RNA had different conformational combinations that allowed self-replication. Recently, Philip Holliger's lab at the MRC Laboratory of Molecular Biology bioengineered an ribozyme that was capable of replicating folded RNA structures. The catch is, it required 3 complementary bases joined together, contrary to 1 complementary base per nucleotide of the parent strand. RNA is also very unstable, and its replication is super erroneous. Some studies conducted in non-prebiotic conditions have shown self-replication of RNA without much external support. But there comes the TAR paradox. That is, every organic molecule ultimately forms TAR. And also larger RNA fragments tend to get shorter, losing information, not very ideal for life, differently. One thing that needs to be mentioned while discussing about RNA world hypothesis is Spiegelman's experiment on the replication of bacteriophage MS2, which has RNA as its genetic material, inside Escherichia coli, the microbiologist's most loved bacteria, which has DNA. The details of what he did might be a topic for another day, but what he concluded was super exciting. He observed natural selection in his flask. For example, some RNA trimmed the genes not necessary for surviving in the test tube. He hypothesized, long before cells were invented, there could have been selective forces that pushed nucleic acid molecules to greater and greater length and complexity in order to solve the selective problems. At the end of his experiment, he obtained what is now called Spiegelman's monster, a short chain of RNA which can only be replicated by bacteriophage Q beta replicase. Recently, some theories like the RNA peptide world hypothesis are getting some traction, which takes inspiration from protein synthesis in cells and hypothesizes the growth of peptide stretches on RNA. Therefore, tries to solve the mystery of co-evolution of RNA and proteins, and why proteins take part in catalysis while RNA stores information. Reconstructing the past is difficult. It is demanding, and that's why it is fun. Understanding who we are has been intriguing philosophers and scientists alike. We went through some theories, challenged ourselves with some fundamental questions, and still we haven't been able to come close to an answer to how it all began. None of the theories are self-explanatory, and probably none of these are fully true. But connecting the dots has been the goal of science and will always be. It is extremely unscientific to tag any of these theories as entirely wrong. Well, perhaps except spontaneous generation. New evidence will come, and so will new theories. Older theories will be modified and our knowledge will be enriched. Scientific knowledge, like life itself, evolves through time. Until next time, think about it. Stay happy and healthy. And remember, in science, even your smallest contribution is extremely important. Also, please like, share and subscribe to the channel. It really means a lot.